good afternoon, everybody, and thank you again for joining this webinar. In this talk, I'm going to present like a brief overview of how the phage-based products are made, the most common products that we produce up to now, the quality requirements for that kind of products, and at the end we will touch briefly also on the regulatory guidelines that are currently available within the European Union framework. I will start with a brief overview or introduction of our company, just to show you how we fit in the context of this talk. Jafral was founded in 2011, and we are an independently, privately owned biotechnology company, and we are located in Slovenia, in Europe. And we are the world's biggest contract manufacturing and contract research organization with the primary focus in development and production of phage-based products for a variety of applications, either human therapeutics, food industry, cosmetics, and so on. We actually have two production facilities, for these both facilities, we have an European GMP certification for production of phages as human therapeutics. And we are required to have actually two different certifications. One is for production of phages as biological products, and that is used when the phages are native phages and are classified as biological products. And the other one is in European Union, at least, phages that are genetically modified are classified as ATMPs, so advanced therapy medicinal products. And for this, we have a separate EU GMP certificate. And in addition to certification for production of human therapeutics, we have also separate certification for GMP production of phages as food supplements. And we also hold an ISO 14001 certificate for environmental management system. So the type of products that we can work with, either phages or hosts, are biosafety level 1 or 2, either if they are non-genetically modified or genetically modified. Phages for a different kind of applications. The most common are for human therapeutics, as is lately, for clinical trials, for compassionate use cases. Then for food supplements, phages are used as probiotics. For animal fat as feed additives, we also produce phages for cosmetics, if they are mixed with creams or serums. And in agriculture, for a treatment of plant diseases. So in general, the variety of fields where phages can be used is quite broad, so they have a lot of potential if the field grows. So we also summarized here, at least based on what our experience are, since we are a contract manufacturing organization, the most common phages that were requested that we produce by our clients are summarized here in the left side. So usually these are phages that target Escherichia coli, Pseudomonas, Staphylococcus, Mycobacterium, Salmonella, Clostridium, and so on. So these are the phages that were up to now, at least according to our knowledge, most commonly used. And then we have also some other exotic phages, but the one on the left are the most frequent. So how does the development of phage product goes? So initially everything starts with the determination of bacterial infection we want to target. And once that is defined, we move to a phage selection process, and where we select one or more phage candidates that can move to a small-scale process development work. And we did that within that initial process development. We always encourage our clients that we produce some small volume purified phage material and put that on stability program, because stability is one thing that cannot be fastened. So it's better to start with the formulation studies and other stability work from the beginning, or at least as soon as possible. And then when the initial process development work is sufficient, then we can also move to scale up activities. Again, we encourage the clients to focus also on the stability, because that is a very important topic. And then if the scale-up is successful, we then move to routine production, either GMP, non-GMP, and again, place everything on the stability program. So in parallel to all the process development work, we need to also develop all the supportive analytical methods that then support production process stability, release of the material during later activities. So everything starts the same as the process development with analytical method development, and what kind of methods needs to be developed are then linked actually to the final product for what it will be used, so the application, because that determines the specifications that the product will need to achieve, and that specification then include all the test methods that will need to be done in order to say that that product is has the required quality that we want. So after the method development is then successful, then we move to method qualification or method validation, and then use that kind of methods where we show that it's suitable for its intended use, then also for routine testing. So we actually prepared, that is really a brief overview of how phages are manufactured. Something was also said on previous talk, but here it's more schematic maybe. So everything starts with the initial selection of phage and the host, and we call that the research phage banks or research cell banks. And here on the left side of the slide, that is a GMP path, how in the GMP environment phage products are produced. So once the research banks are made, we then move to the GMP suites to produce master phage banks and master cell banks, bacterial banks. And all these banks needs to be characterized and placed on stability, and then once their suitability is confirmed, we can then move to later stages of production. In general, in initial stages of clinical trials like phase 1, phase 2, working phage banks or working cell banks might not be requested by the regulatory agencies, but in later stages, usually they are requested to be produced also in the GMP environment. And from that, from when we have both starting materials, we can move then production of the purified phage, that drug substance or active pharmaceutical ingredients. If we are at the beginning of the clinical trials, or we first produce engineering runs that are done in the GMP environment, but cannot be used for the clinical for human use, but are used as a supportive stability testing, for some additional analytical development, or validation of the methods, or formulation studies, and so on. And once the engineering runs are successful and the quality that is predefined is achieved, we then move to production of clinical material, first drug substances or APIs, and then production of drug product if it's a monophage or a cocktail of multiple phages. So that is a regular GMP path. That is the most stringent one and the most time and resource consuming. For the other applications that are not used for humans, then this production process is a little bit more simplified, but still everything starts with a selection of banks, phage and host, production of banks that are also characterized so that we know what the starting material is, and then production of purified phage and the final product. So that is like a brief overview of how the manufacturing process works. So here are some examples of how the final products are then packed. We detect in liquid, either in bulk from 5 milliliters to 20 liters. We can pack in liquid in vials, like aliquoted for single use or multiple use, in various milliliter sizes. And in addition to liquid, also phages can be lyophilized or spray dried to obtain a dry powder, which can be packed in vials or encapsulated in capsules for oral application. Okay. At the end, when the phage product is produced, quality control testing needs to be done. Again, the selection of the methods that are required are based on the application for which that kind of product will be used. In general, for the intravenous application, the quality control is the most stringent and includes a lot of tests, but usually appearance, pH, some kind of potency to determine how many active phages are in the product are done. Identity testing and sequencing is lately a must. And then we have all the safety tests like endotoxins or microbiological quality. And again, it depends on the process and how complicated the process is. We also need to test for impurities, either if there are residual impurities from the host, like host cell DNA, host cell proteins, or some kind of process-related impurities if the antifoam is added to the process, or some kind of endonucleases, then it might be required to test them also in the final product. And here are also the examples, how this would look like: an appearance test, a potency test on plating on the agar plates, electron microscopy, particle counts with DLS, and so on. Okay. So what we wanted just to show at the end of this presentation is the current regulation or guidelines within the European Union. So as they are currently for the preparation of phage products for human therapeutics, in a GMP standard, we need to follow the EU GMP guidelines, EU X Volume 4, Part 1 and Part 2 for drug products and APIs are linked to biologicals. So if we produce native bacteriophages, we are following that guidelines. If we produce genetically modified bacteriophages, we are following Part 4 for the ATMP or advanced therapy medicine products. And maybe just to add a comment here, in 2023, in European Pharmacopoeia, they issued a chapter for the phage therapy medicine product that is 5.31. And this is, it offers a framework of the requirements of the active substances and drug products for phage products for the phage therapy. And as was said in the previous presentation, also in the 2023 issued a guideline for the veterinary medicinal products. So this was really a brief overview how the phage manufacturing works and what are our experience. So if you have any questions, please let us know.